All right.
Hi, everyone. Thank you for joining us today. I'm very excited to share our research that I just celebrated the 5th year anniversary at UC Davis. And if someone can confirm that you can see my screen and hear me well, that would be great.
Yeah, it looks good.
Great. So we are doing biometeorological measurements, modeling, and remote sensing with hope to have real world impact. And this work is based on work on many people in our lab and UC Davis and UCNR collaborators. So thank you, everyone. I'll be happy to receive your feedback after this review of our work, but also you feel free to interrupt me at any point if you need any clarification. So in order to do this statewide program, we started this observational network between different crops. California has many crops and management options that we needed to monitor to develop some information on local values, but also to have, I need somebody to mute. Oh, cool. So we also need to address the water use of these new management practices that are becoming, some of them for drought resilience, some of them for climate change impacts. So these are some of the landscape we've been measuring and where they are in the state. We do have to choose the option of evapotranspiration measurements, because in order to have this large network of observations, we are using the residual of energy balance. So we oftentimes can't afford infrared gas analyzer and then get evapotranspiration as a residual of surface energy balance. And this method is a little bit more generous toward ET because of the famous problem of eddy covariance having troubles closing the energy balance. We take that lack of energy balance closure and put it all into latent heat energy. So ET values are a little bit elevated, but we think they are suitable for on-farm water management, remote sensing validation, and validation of different models. And we also do sometimes have to simplify our measurements to use surface renewal measurements where sonic anemometry will be skipped. And also sometimes we can just focus on daily values and assume that soil heat flux will be 0 on a daily basis. One of the examples of our field setups is in rice in Sac Valley, and we actually study these fields only when rice is not grown. So with the drought increasingly affecting California water, we know that there are oftentimes decisions that farmers make in rice fields to fallow or have non-irrigated cover crops. And sometimes these are not even related to the drought, but the organic rice production, where it's needed to have the rotation in order to manage better for the weeds. So we try to do above ground measurements of evapotranspiration, inputs of precipitation, also levels of water table and soil moisture, so that we target as many water budget components as we can to see how the non-irrigated cover crops in these winter fields and spring can affect this movement of buckets of water. And also these cover crops are very important for wildlife habitat. So because we are on a Pacific Flyway, these cover crops, even when they dry down, they are a good shelter for nesting wildlife. And if there is a disking for removing those cover crops, those nests can be destroyed. So our goal was actually to see if there is any difference in water losses when the rice fields are fallowed or have cover crops. And our setup look like this. We had the winter wheats, a mix of oats, vetch, and peas. Fallow field, of course, in this high clay content soil, we can see a lot of cracks, but I wanted to show you how that really looks in our experiment. And then vetch is a very popular cover crop in this sense. And our results over this 3-year study, where we were just doing winter and spring observations, showed that in water transfer part of the year that starts around May 1st, field fallowing or cover cropping have similar values of water use between 4 inches the least in vetch field in 2021 to 5.5 inches in some cover cropped or even fallow. So depends how wet is the year. Do we have cover crops even successfully germinating if there is a water logging and flooding those fields, or if we have the very dry winters and cover crops might be very short-lived and dry in March before the highest water demand from the atmosphere will be affecting them. And I couldn't resist but to show you how many birds we see in these fields. We're very fortunate to work on this experiment and enjoy all the wildlife habitat. Now we got a new funding from Department of Water Resources to continue this study over new 3 years, but this time we will be observing also rice fields where rice is grown as a business as usual, and then we will be again observing bare soil and cover cropped fields through all the operations that happen between different seasons and months. And this time we really want to do this on every day of the year, not just winter and spring, but to see like how on a yearly basis these water buckets move, how much there is a water loss, how much these cover crops can help store some water from the winter precipitation, and where does it go? So it's going to be another 3 years of these measurements and also adding carbon. So we will be observing water and carbon simultaneously and seeing what does it mean for carbon fluxes when we have bare soil and field idling. We have a lot of orchard observations because there are a lot of orchards in our state, and the management is changing. So we are having drip irrigated sweet cherry orchards where we observe 3 different orchards nearby that have different density, row orientation, rootstock. And we want to quantify the water use and develop local crop coefficients which were lacking. So the California Cherry Board supported us in this effort, and we have 5 years of measurements. We are now also measuring almonds that have either bare soil or cover crop in grazing, as new regenerative practices are being evaluated by farmers, but also our colleague researchers. So we want to respond this one piece of puzzle that is missing on water use. We would often think that these cover crops increase the water use, but I'll go into more detail on results, what we see and what our hypotheses are. We do also measurements in olives. They are now super high density olives. We see these walls of olives that there is a difficulty to distinguish one tree from another, and things have changed since they were grown in different systems. So some of our orchards also have cover crops in this particular one that we see, and our measurements will represent the full irrigation, whereas our colleagues Julia and Paula and Ken Shackle are stressing the corners of the orchard that are outside of the footprint of our measurements, and these are carefully stressed in periods when it could be beneficial actually to improve the olive oil quality. So based on full irrigation management that we measure in the middle of the orchards, they are experimenting with the different stress levels. Something more on the sweet cherry experiment. These are the 3 orchards I mentioned. Our student Jarin Tasnimanika is helping us by running eddy covariance towers, closing the valves in post-harvest to see how the regulated deficit irrigation can impact the maybe some water conservation, and making sure to take harvest samples to evaluate which stress levels in sweet cherries will not affect the quality or quantity of the yields. There's some more information on these orchards, and we have towers in each one of them. Now that we have 14 season year site observations, differently from the past where we didn't have that opportunity to measure in so many sites and over so many years, we can see how much variability there is when one tries to develop crop coefficient curves. This is in green dots, you can see the average value of this 14 season years, and you can see how much variability there is. So I asked Jarine to compute actually what is the maximum difference we see in this modeled crop using this universal crop coefficient values versus measured for each site and year, and maximum pre-harvest difference was 90% cumulatively. And then we saw that there could be post-harvest difference as well. And if we take into account whole growing season, there could be 20% difference. So we'll see how that relates to when we intentionally stress the crop. These are some of the pictures of our towers. Jarine taking the harvest samples even on holidays where growers didn't expect her to show up at 5 a.m. And these are some of the lab evaluations she's doing. If we now compare the stress in post-harvest season to what we get in harvest analysis, we can see that dark blue being the baseline of stem water potential, we see that we have even the fully irrigated drip lines in this light blue will show some values that are not the targeted baseline values, but if we observe this dashed vertical line where the stress treatment started, there is definitely stress observed in these pink values of the stressed trees. And actually, we did see the yield quantity was for around 40% less in the stressed row on average, but one-way ANOVA showed that there wasn't a significant difference because there was so much variability between the sampled trees. And other parameters of the yield quality did not show significant difference either. Jarine did this analysis after 24 hours and after 1 week after taking the samples. The site number 2 has shown much less stress in this yellow color that is in post-harvest deficit. And there was more yield in that orchard where there was a post-harvest deficit. However, the stress wasn't very strong in this case, and we didn't see significant difference either in harvest quantity or quality. The site number 3, we see this dark orange stress tree. The stress started even before our closing the valve, so it was already tree that might have been affected, and we see that around 30% difference in the yields, again without having significant difference. So last year after the harvest, we stressed the same trees again and in a longer post-harvest period, and we hope to see how much this will affect the yield in the spring sampling when Jarine goes again to analyze these cherries. Water savings we had in post-harvest period, we had anywhere between 26 and 50% of water savings. If we take that value on the seasonal basis, that's between 10 and 37%. That could be water conservation if we do stress cherries. And if we don't have any effect on the yield quantity or quality. Another experiment is in almonds, where we had sites with bare sore almonds and the cover crop, again, trying to quantify above ground evapotranspiration losses and also what's happening in the soil, runoff, and precipitation. So various inputs and outputs, and seeing how the cover crops might be beneficial in storing that winter precipitation instead of through runoff. We had Marga, who just graduated and got a job in Sac Valley. And we have Ian and Jareen working on this experiment to continue Marga's work. These are the sites we have. They are kind of concentrated in a similar area of California. And we have this black dot is the tower we're installing later this week, so that we have at least cross valley gradient if we couldn't scatter the towers in the larger almond growing area. But we did find the sites that are fully bare soil every season of the year. We have a site that has some residual cover. It's still conventional, but has a rich seed bank. We have cover crops where the grower is bee friendly, having some nice seeds for bees, tall cover crops. And then we have seasonally grazed and cover cropped orchard where it has a very high regenerative score. And then we have farmers that do off-ground harvesting and all year grazing. And we are adding this site, which will be also seasonally grazed with drip irrigation. All these sites have sprinkler irrigation except the bare soil one that has drip irrigation. And now this new site will also be interesting addition to have drip irrigation with cover crops. So those cover crops in alleys will dry sooner than when we have sprinklers. And if we take 17 months of our measurements and see the average values wherever we didn't have large data gaps, and see the monthly, see the monthly averages, we see that the bare soil almond in both years had some of the highest water use, and these other orchards that have different degree of cover crops all had a lower water use. This light blue and dark blue are maybe the most fence-to-fence comparisons because they're close together in Gustine. They're managed by the same grower. And we also saw the lower, a little bit lower values in evapotranspiration. While we had the measurements, our collaborators needed to remove this equipment, so we don't have enough measurements, but we're starting them again. So stay tuned for more results. And I wanted to also draw your attention to this values in parentheses. We take these as a measure of how regenerative is each site. So the higher the value, the more regenerative they are. And then that's useful to explain this plot where we tried also to see what is the water use efficiency if we take how much there was crop per drop. And in our case, in the orchards we selected, the higher the regenerative score, the less crop per drop we got. However, these two most regenerative sites are actually having root disease in one case and stunted trees in another case. So maybe it wasn't very fair comparison between the sites, and maybe these two would be also close together to compare. However, this light orange site has very old almonds that are at the end of their production cycle, where this is 14-year almonds at probably at their peak of their production. So maybe not very fair comparison, but something to think about. Again, we have a very good data set on soil moisture with the soil profilers that go down to 100 centimeters or 40 inches. We have 9 depths of measurements every hour of the year. So in order to try to present this information in a meaningful way, we decided to divide it into irrigation and non-irrigation seasons, so that we see how much is it that water is given artificially through irrigation, or how much it is supplied naturally through precipitation, and which systems were more efficient in storing it. So on average, we see that the bare soil almonds that are this dark orange were more heavily irrigated and had more moisture in the top foot of their soil layer, and had not so much water down the profile, whereas more regenerative that are bluish or greenish had more water storage down the profile, and it seemed that they had less irrigation supply. In non-irrigation period, where we had the very wet winter, now we see that these bluish and greenish orchards had much more storage down the soil profile and were more efficient than these dark orange that was less efficient. Some of our colleagues that are more experts on soil hydrology have shown this before. We're just confirming with these soil profilers we have. Also, we're not experts on the soil organic matter, but we were taking 8-foot deep soil cores in 3 alleys and 3 rows within the footprint of our tower in each of the orchards, mostly to see how the water storage changes on annual basis. But these soil samples were also useful to see the soil organic matter. And again, if you see the higher regenerative scores, this Gustine and Merced that had the highest regenerative score had the highest organic matter. These are grazed and cover crops. Something that other researchers have shown, and we confirm and will continue sampling as we continue this experiment. Also, because we are unable to cover a large almond growing area in California with our ground measurements, we have a postdoc and a project scientist that are working on upscaling this using the remote sensing and modeling approaches. We have Yifang Wan working on OpenET comparisons to our ground measurements, and she got these results for cumulative annual water losses from these conventional orchards that we observed has the higher water use. It also had the highest water use when remote sensing products are used, and she has just started this month, so stay tuned for more results on remote sensing comparison to our ground measurements, and we will be evaluating different products, remote sensing products, also different models that enter into OpenET, as well as ensemble values that you see here. And then Dr. Dave Piles is using his methods ACASA for land surface modeling, and he's slicing the soil canopy and above canopy atmosphere into numerous layers to model the exchange between different layers. So he has just started working with us as a project scientist and will be contributing more results to upscale our ground measurements. Another question that we got after starting to address the water use of cover cropped almonds was, do cover crops also increase the frost risks? So we called Mark Battany that has this tiny self-logging sensors that were very affordable and easy to deploy at almond tree. So we had a grower collaborator that did this herbicide applications to make the blocks of his orchard under cover crops and others to be bare soil. So we installed multiple sensors and monitored for one winter season in 2022 how cover crops might impact the almond temperature at the bearing branches height. Around 3 feet would be the height we deploy them, because we know every layer above that will be warmer. So we just started where the almonds start to form and where the coldest temperatures can impact them the most. And then we had Emma and Ian work on this data set, and we had a camera that shows the first day when we deployed the sensors, the last day of our observations, where we also can analyze averages, 50-minute averages of differences of nighttime temperatures between cover cropped and bare soil blocks of the orchards. And we did this just for nighttime because these little sensors were not shielded, so we couldn't counter their daytime measurements being reliable. So the nighttime is also when the frost occurs. We see that in month of February, more these reddish colors where the cover crops impact the temperature to be higher in the orchards. And then as we are transitioning through more cover crop growth and leaf out in almonds, we are getting lower temperatures where cover crops are. This one distinct night of reddish color, darker reddish colors, is when we had this severe frost in 2022, and the grower sprinkled over the cover crops and bare soil orchard blocks to protect against the frost. And actually that was our hypothesis, that if we have cover crops in the orchard, we have more roughness surface and closer to almond trees to form that ice and keep the orchard safe from frost. So in order to continue from these several blocks within one orchard, we equipped same measurement setup in now full orchard blocks where we have eddy covariance towers. And since we did that, fortunately for farmers, unfortunately for our study, there wasn't really frost forecast for them to irrigate. So we couldn't test our hypothesis, but we could confirm that cover crops do lower the temperatures. These blue ones are bare soil almonds, and the red one, these blue ones are cover cropped orchards, and the red ones are control. So cover cropped would lower the temperature even below the critical temperature for almonds when there is a forecast for frost. So something to be careful about. And then one of our new projects that we started last year is in the Delta annual crops, and this was funded from Delta Conservancy in the state for drought relief program. The farmers that participate and collaborate with us to host our experiments had either field fallowing or cover crops that are non-irrigated or irrigated ones to just germinate the seeds. So we see these different systems where, for example, safflower, that is a very deep-rooted crop, was had a very successful stand in this non-irrigation management. We had non-irrigated pasture. We had deficitly irrigated corn that is for silage. So very different systems of the different Delta islands. And we had a fallowing field actually with very high natural vegetation because it's close to the river and its water table is quite high. So another experiment where we will be doing farm scale measurements across different Delta systems to go from plant and farm scale to satellite observations to evaluate them, help evaluate different models. And our initial results didn't show very good agreement, but these are off-season values that Emma and Olmo are working on, and we will have 3 years of data through different seasons of the year to show how different crop growth can be more matching or mismatching what we see from the satellites. And some of the ground measurements, if we take these different systems to compare them to each other on August to September basis, we took average daily value for evapotranspiration and photosynthesis, and we can see yes, when we have maize crop that, although it's low irrigated, it was able to develop quite a bit of biomass, and we see more water losses also from our natural vegetation that was supposed to be our fallow field. It had a lot of vegetation in this period that we are observing since last August. And then our fallow field is actually post-harvest winter wheat, and rain-fed pasture was pretty dry in that period. So we see higher water use, of course, where we have larger biomass, and then we see higher carbon uptake as well, and higher nighttime respiration in these fields. So it will be very interesting to simultaneously analyze water losses and carbon losses or uptake, so that when we develop information on some drought management, we know the cost of these practices also in carbon, especially in the delta where these islands are so vulnerable, and we need to keep that carbon in the ground as well in these systems. We also have Dr. Dave Piles here working on modeling these values and using some previous experiments and building up on that work to model all these fluxes and also have them upscale to larger delta area. We are also working with DWR and evaluating the footprint of semi-stations so that we can help the state in that sense. If there are new semi-station locations, to know what is the minimum of that irrigated grass surface that is needed. We know it's always necessary to expand that network of semi-stations, but also there are large limitations in how much land is available. So Chitra and Olmo are working on a footprint of this semi-station so that we know better which are the grass surface we need to have reliable values on reference evapotranspiration. Olmo is very passionate about improving surface renewal methods, and as I mentioned earlier, it's a method that has a reduced equipment and is more affordable so that we can have more ground observation. And our newest project is in Redwoods. So we got funded by the state and specifically Cal Fire to install 2 tall towers in Redwoods state forest. We will be observing from the ground again from these tall towers what's happening in early and mid-seral stages of the forests to inform better the remote sensing products and see how the redwood forests are doing under changing fog. There is some evidence that the fog is declining, and we know the redwoods are very dependent on fog because they have very poor stomatal regulation and lose a lot of water when they're fog. And also we want to see the carbon sequestration. We know these are very highly productive and systems that are sequestering a lot of carbon. We know they're sink for the carbon. We need to put the number what that is, and because 93% of the forest is second growth, young and mid-seral, that's where we will be installing towers, also because we need to pay attention to the height of the tower not to be affecting the air traffic. And this is going to be very strong observatory with 100+ variables measured in real time and continuously. And we have funding to do this experiment until 2026, and we are actively seeking collaborations and new ideas to try to keep this as a long-term observational sites to have really meaningful results if we can measure for 10 or 15 years what's happening. And the collaboration team is large. Halcyon and I are leading it from Lawrence Berkeley Lab and UC Davis, and our colleagues are giving us a lot of support through co-PI-ship, and we're hiring some new people to make this happen. Thank you so much. I'll be happy to hear what feedback you'll have and how we can collaborate more on these projects or something new.
Thank you very much, Dr. Sovočarev, for your presentation. And now, we have time, quite about 25 minutes for questions. Just go ahead and ask directly. Thank you. Questions?
Hi, Steve.
Hi, Kosana, how are you?
Excellent. How are you?
Nice seminar. You mentioned drop per crop. You didn't talk too much about yield issues associated with water use. And what the cost of those practices were in terms of the efficiency is water use efficiency, right? In terms of yield. So how do those various practices in the orchards, for example, rate, or maybe you don't have enough data yet, didn't say.
Yeah, exactly. Thanks. Yeah, that's a very good point. We need to collect more yield data. So far, we are just using water use efficiency in almonds as crop product, where we had farmers report their yields. I guess we can do that in cherries because Jarin is collecting this nice data set and samples, but also for us flux scientists, water use efficiency can be also carbon uptake per water use. So there's another water use efficiency term that we will be able to express once we have annual carbon balances in our data set, and we're just starting installing those sensors that directly measure evapotranspiration and simultaneously give us carbon values too.
Any other question?
I have a question.
Sure.
Kosana, how do you decide where to put the soil sensors when you're making those measurements in drip irrigation?
Oh, yes, we put the soil sensors, I guess you're talking about almonds.
Yes.
We put them in under the drip line in the tree row, and then we put one in tree alley on one side and one in the tree alley on the other side. Unfortunately, they're not standalone sensors for us to scatter them around the orchard, which would be more beneficial. And we have to have them close to a tower. So because there could be large variability in cover crops in the tree alleys, we put them on each side. So two in each alley, and then one in a tree row to see where the irrigation is supplying water at which depths, from which depth are almonds using that water, and then also similar for cover crops, how much they can help more water infiltration in the winter, and for which depth they use that water. Do they like store more than they use?
Okay.
Yeah, thanks. Very good question.
Okay. I have a question on my own. I was wondering how do the ET measurements that you are ascertaining in your fields compare with the data from OpenET and other remote sensing tools currently available? I'm asking because a number of growers asked that kind of questions. I was wondering what you found out from your research so far.
Yeah, so it's variable, and we are very interested to see how much these different management practices, like the picture you see in the background is so dry. We wonder how much there is we didn't see a very big agreement. Then when we have bare soil almonds, we see higher agreements. We just started with our remote sensing person to do this evaluation across 20 towers that we have, measuring such a different landscapes, including redwoods.
Okay, thank you. Any other question? Oh, go ahead, Stephen.
Since no one else is raising their hand. I was interested to see the infiltration data that you had, where it seemed that water infiltrated, or more water infiltrated deeper into the profile where you had a cover.
Yes.
Is that necessarily a good thing? We talk about leaching losses, and was water infiltrating below the root zone of the almond? I mean, how does that relate to almond production effectively? That infiltration data and profile saturation data.
Yeah, because I'm not an expert on below ground soil, I took from my colleagues that this is that the cover crops actually decrease the nitrogen leaching, although they could store more water. And we're taking the samples down to 8 feet, like we did in safflower and sugar beet experiments with you, Steve. We are taking, but we didn't see that we could have a meaningful results in this beyond root zone 4 to 8 feet depths. So Jareen is working on analyzing those, but maybe you can help us with that. We're not sure we're seeing significant differences. And then I guess we could also analyze for nitrogen in these soil samples we're taking to see if it's leaching more or less when we have grazing and the cover crops. But definitely we see that they do help water.
You know, we talk about this notion of regenerative agriculture, which presumably is a positive thing, but that would be an interesting question in that context, it seems to me.
Totally. Thanks, Steve. Yeah.
I have another question.
Yes.
The CIMMYS project, where you're doing the fetch around the CIMMYS stations, have you collected data yet?
We are collecting the data and preparing the final report by the end of May. We started with measuring with eddy covariance to compare reference ET to measured ET, and now our student Chitra also installed the temperature relative humidity sensors, like 9 of them across that CIMMYS grass to see in a scalar sense how much there is a difference between the sensors that are closer to the edge and that are further away, and we do see some differences. So
Have you looked in different microclimates or just one location?
we're just doing it in Davis at one location.
Okay.
Okay. I don't know if anybody, I mean, we still have lots of time, more than 10 minutes to entertain a number of questions, but in case nobody else has an additional question, I'd like to thank Dr. Sovočarev for honoring. Oh, we have a question again. Stephen, please go ahead.
Yeah, I mean, this is a great opportunity.
We need to meet soon.
That's true. There's no reason to think that a sown cover crop or an abundant weed cover would have any functional difference, is there? Around here, for example, and where we live in Davis, all these orchards have abundant weed covers. There may be control under the tree rows, but basically, lots of mustards and radishes and grasses and malva and various things like that are growing, and then they're mowed. Of course, this is north of the Delta, so where there's more winter rainfall here. So just a question.
I didn't understand what's the question.
Is there any reason to think that a sown cover crop would be any better or different than a natural weed cover crop in terms of these properties of interest and regenerative definitions?
Yes, so I know that Project APSAM has a seed for bees that are carefully selected plants that favor pollinators habitat, and then we have they also have carbon building mixture. So I don't know, the variability between orchards is already large, and we don't have very good control to compare for water. The vegetation type would be too difficult for us to know from what we measure from the towers that mixes whole system, almonds and cover crop and soil together. So I think that's a question for somebody else that can do more science at the tree alley level than us, that we're doing it from the towers as a whole system.
Thanks.
Thank you, Steve.
Okay, yeah, once again, Dr. Sovočarev, thank you very much for honoring our invitation to give this talk. And yeah, anybody who has further questions could reach you to your work email. Thank you all very much.
Thank you, Tobias, for inviting me.
Bye. Bye, everybody.